Assalamu alaikum. Welcome to my channel Pharmacology Made Easy. In previous session we'll discuss about insulin structure, insulin synthesis, insulin storage. Now today's session is all about insulin release, the mechanism of secretion of insulin, and also the regulation or the factors that are responsible for insulin secretion and insulin inhibition. So at first I welcome you to my channel Pharmacology Made Easy, and thank you all for listening this conversation. So I start the conversation. In previous session we'll discuss that insulin is an anabolic hormone and its molecular weight is 5808, and it is the only hormone that control our blood sugar level, and there are many other hormones that increase our blood sugar level. So insulin is secreted by the various stimulus, and it is secreted from the pancreatic beta cell and helps to control our blood sugar level. The insulin is synthesized from the rough endoplasmic reticulum of the pancreatic beta cell, and it is processed and packaged into the Golgi apparatus, and it is stored in the secretory vesicles. In the secretory vesicles, the 6 mg of insulin is bound with 2 mg of zinc, and total content of insulin in our pancreatic beta cell is 8 mg, that is equal to 200 international unit. From this, 25 to 30%, that is only 25 to 30 international of unit of insulin, or some authors say 30 to 40 international unit of insulin, that is secreted on a regular basis. So how insulin is secreted, and today's concerned topic is about how insulin is secreted, what is the mechanism of insulin secretion, and what are the factors that are responsible for activating this or inhibiting this process. So insulin is stored into the pancreatic beta cell secretory vesicles, and insulin is stored as because insulin is synthesized from its precursor, that is preproinsulin, that is after that it is converted into the proinsulin, then it is converted into insulin. So insulin is stored into the secretory vesicles of pancreatic beta cell. So in the pancreatic beta cell there is a transporter that is called the Glute transporter, in which when there is excess glucose we have taken, the pancreatic beta cell taken up the glucose into its cell or into its cytoplasm by this Glute transporter. There are many Glute transporter, that is 1 to 5, and Glute transporter especially present into the pancreatic beta cell. If we say a dialogue that is PQ likes Glute2, that is PQ or pancreatic beta cell contains the Glute2. The Glute2 also contains that PQ likes. Here L is liver, and liver is responsible that hepatocyte contains the Glute2. Like LIK, and the I is responsible for intestine, and intestinal cells contains Glute2. LIKE, that is the K, and K the kidney, the renal cell contains the Glute2 transporter, and also the enterocyte contains the glucose transporter. So these are the sites that the cell contains the Glute2 transporter in their cell surface area. So through the GLUT2 transporter, the glucose enters into the pancreatic beta cell, whereby the glucokinase enzyme, the GLUT2, the glucose is converted into the glucose 6-phosphate. This glucose 6-phosphate become glycolized by glycolysis, and the glycolysis of glucose 6-phosphate, it is converted into pyruvic acid. This pyruvic acid enters into the cell mitochondria and goes into a TCA cycle, and also oxidative phosphorylation occurs where they convert the ADP into ATP. This ATP is highly sensitive for potassium channel. When this ATP blocks the potassium channel which is present into the cell surface, normally the pancreatic beta cell contains a potassium channel, and this potassium channel is present into the pancreatic beta cell, and the resting membrane potential of pancreatic beta cell is minus 70 millivolt, in which the potassium is kept out of the cell and maintain the negative polarity into the cell, and that's the resting membrane potential of the pancreatic beta cell is minus 70 millivolt. When there is more ATP synthesized, when GLUT2 transporter entry more glucose through glycolysis, it converted into the more pyruvic acid, and more pyruvic acid is converted through TCA cycle and oxidative phosphorylation. They produce more ATP. When there is more ATP, these ATP sensitive potassium channel will be blocked, because more ATP blocks the potassium channel. When there is blocking of the potassium channel, the cell positivity increased into the cell, because potassium is a positive ion, and this positive ion is increased into the pancreatic beta cell, and the resting membrane potential become minus 50 millivolt due to positivity of the potassium into the pancreatic beta cell. Due to the positivity of the potassium into the pancreatic beta cell, as far the ATP sensitive potassium channel is blocked, their inside increase the depolarization of this pancreatic beta cell. When there is depolarization of the pancreatic beta cell, the cell positivity is more increased, and this negativity of the cell becomes positivity, and the minus 70 millivolt of resting membrane potential becomes minus 50 millivolt. So there is depolarization start into the cell. This depolarization causes opening of the calcium channel into the pancreatic beta cell. The pancreatic beta cell contains another channel into its cell surface, that is the calcium channel. When there is more depolarization and more calcium enters into the cell, because the extracellular fluid contains more calcium and intercellular fluid contains less calcium. So from the higher concentration to lower concentration, the calcium enters into the cell very easily, and this calcium enters into the cell by depolarization, causes EXocytosis of the insulin from the secretory vesicles from the cell into the outside. So insulin is released due to the depolarization of the pancreatic beta cell. As far there is increase of calcium channel, this calcium enters into the pancreatic beta cell through the calcium channel and causes the secretion of secretory vesicles insulin from the pancreatic beta cell into the outside through EXocytosis. So this is the secretion of pancreatic beta cell. So it is very important for us, because many regulatory hormones and many regulatory factors that are responsible for increase the insulin release through the pancreatic beta cell by this mechanism. So what are the regulatory factors that increase the insulin release from the pancreatic beta cell? At first, there is high amount of glucose. When there is high amount of glucose, the glucose is more converted into the glucose 6-phosphate, and more glucose 6-phosphate converted into the pyruvic acid, and this pyruvic acid converted more ATP. This more ATP inhibits the ATP-sensitive potassium channel and more depolarization of the cell. That's why the calcium channel becomes open, and the calcium entry of calcium causes more release of insulin. So excess glucose in case of hyperglycemia, in case of excess glucose intake, there will be more insulin release from the pancreatic beta cell. Second, more intake of protein that causes increase the high amount of insulin from the pancreatic beta cell. The third, the special amino acid sum, that is arginine, leucine and lysine, that also increase the release of insulin from the pancreatic beta cell. There is another hormone that is called the incretin, that is a gut hormone that is stimulated or released due to oral glucose intake or postprandial glucose induced the gut hormone, the incretin. The incretin also increase the glucose induced insulin secretion, and these incretins are mainly GLP-1 and GIP. The insulin is an important gut hormone that is released after taking of glucose or excess amount of glucose by orally. Only oral glucose goes to the intestine, it stimulates the gut, and the gut will release the incretin. Incretin is a group of hormone that is contains the glucagon like peptide 1, that contains GIP or glucose inhibitory peptide, or another name of GIP is glucose dependent insulinotropic hormone. This incretin also the incretin is the secretin, the cholecystokinin and the gastrin. These are also incretins. So insulin is a group of hormone that is released by oral glucose intake, and it releases from the gut wall, and that causes the stimulation of the pancreatic beta cell to release the insulin. So these are the important factors. Some drugs also causes stimulation of the pancreatic beta cell and causes the release of insulin. These drugs are sulfonylureas, and sulfonylureas are composed of first generation and second generation. The chlorpropamide, the tolozamide are first generation. Second generation is clibendelamide, glipizide, glicazide. These are the second generation of drug, and these drugs, the sulfonylureas, causes stimulation of the pancreatic beta cell and causes the release of insulin by this mechanism. Not only sulfonylureas, the oral antidiabetic drug, the meglitinide and diphenylalaprotide, that's are ripaglinide, nataglinide. These drugs also acts by stimulation of the pancreatic beta cell to release the insulin. So these are the important drugs and these are the important factor that causes the insulin release. There is some the other factors that inhibit the insulin release. That one is low containing of fatty acid. This low containing fatty acid inhibit the pancreatic beta cell to inhibit the release of insulin. Some drugs that causes the inhibition of insulin release from the pancreatic beta cell, and these drugs are called important drugs that are diazoxide, colchicine, phenytoin. They inhibit the secretory vesicle of the pancreatic beta cell, or they inhibit the pancreatic beta cell to stimulate or release the insulin from them. And this diazoxide is an important drug that acts by activating of potassium channel. When there is activation of the potassium channel in the smooth muscle and in the pancreatic beta cell, this diazoxide stimulates the or activation of the potassium channel into the pancreatic beta cell and release of potassium from the pancreatic beta cell. That's why there will be no depolarization into the cell, and there will be hyperpolarization of the cell, and there will be no calcium entry into the cell, and so there will be no release of insulin. So in this insulinoma, when there is a hyperplasia of the pancreatic beta cell, that causes more release of insulin and that causes prolonged hypoglycemia. In such patient we give diazoxide, and diazoxide is an important drug that acts like that. So diazoxide, colchicine, phenytoin, these are the important drugs that inhibit the insulin release from the pancreatic beta cell. There is another hormones that inhibit the release of insulin secretion from the pancreatic beta cell, and these hormones are quite related with some disease also that inhibit the insulin release, and that's why they produce the type 2 diabetes mellitus. And that's one hormone is glucagon, in which in glucagonoma there is more production of glucagon, and glucagon causes in the liver neogluconeogenesis and increase the glycogenolysis and increase the blood glucose level, and this blood glucose level is more high, and this blood glucose level causes more insulin secretion, but the insulin are not secreted as so high as they control the blood sugar level. So that's why there will be chronic hyperglycemia occurs, and this chronic hyperglycemia leads to type 2 diabetes mellitus. In case of growth hormone, there will be hypersecretion of growth hormone in an adult, that is called the acromegaly. In acromegaly there will be also neogluconeogenesis, and there will be glycogenolysis, and the blood glucose level is highly increased. This highly blood glucose level increase causes high release of insulin from the pancreatic beta cell, but stimulation of the pancreatic beta cell, the beta cell become exhausted after some times due to hyperstimulation of pancreatic beta cell due to chronic hyperglycemia. And when the pancreatic beta cell become exhausted for days by days, there will be no release of insulin from them, and so chronic hyperglycemia cannot be controlled by the presence of insulin in the blood. So there will be type 2 diabetes mellitus. In case of cortisol, if we say Cushing syndrome, there will be also neogluconeogenesis in the liver, and gluconeogenesis in the liver, and glycogenolysis that produce in glucose in excessive amount in the blood, and these also stimulation of the pancreatic beta cell to release high amount of insulin, but this stimulation causes again exhausted the pancreatic beta cell, and after some days it cannot secrete more insulin, and this insulin cannot control the blood sugar level in chronic hyperglycemia in Cushing syndrome. So the glucagonoma, acromegaly, and the Cushing syndrome, they all may causes the type 2 diabetes mellitus. So these are the important regulations of releasing or inhibiting the insulin, and it is important to us to think this kind of thing, and also understand these things. Hopefully we understand these things. There is another thing that we should discuss on this video, that is the glucose transporter, Glute transporter, where the glucose is enters into the pancreatic beta cell by the glucokinase. It converts into the glucose 6-phosphate. When the glucose is converted into the glucose 6-phosphate, it will not be out from the cell, and this glucose become trapped as the glucokinase converted into the glucose 6-phosphate. So by Glute, the glucose enters into the pancreatic beta cell and converted into the glucose 6-phosphate. So this conversion of the glucose into the glucose 6-phosphate cannot out of the glucose from the pancreatic beta cell. So after entering into the pancreatic beta cell, glucose cannot be out, it is become trapped into the pancreatic beta cell. So our concern thing is about glucokinase. This glucokinase enzyme, that is the rate limiting enzyme that converts the glucose into the glucose 6-phosphate, sometimes it becomes mutant, or sometimes it becomes there is mutation of this glucokinase enzyme. So this mutation of the glucokinase enzyme, the glucose cannot be converted into the glucose 6-phosphate. So inside the pancreatic beta cell there will be more glucose, and more glucose into the pancreatic beta cell causes less insulin secretion, because there will be no conversion of the glucose 6-phosphate, and this glucose 6-phosphate cannot be converted to pyruvate by glycolysis, and no pyruvic acid is formed, so no conversion of the ADP into ATP. through the TCA cycle and oxidative phosphorylation, and so there is no blocking of the ATP-sensitive potassium channel through the excess ATP, and that's why there will be no depolarization of the cell, and there will be no calcium entry into the cell and no release of insulin. So this mutation of the glucokinase occurs in young patients, and they produce no insulin release, and there will be chronic hyperglycemia, leads to type 2 diabetes mellitus. In a young patient, we know they are suffering from type 1 diabetes mellitus, but in this condition, due to mutation of the glucokinase or the or deficient of glucokinase, this type 2 diabetes mellitus occurs in young patient. That is called MODY, mature onset diabetes of the young, or monogenic diabetes of the young. So this glucokinase is important enzyme that is responsible for causing the type 2 diabetes mellitus young patients. So thank you all, thank you your perseverance and patience for listening to the video, and if you like this video, please share, comment and subscribe my channel. So thank you all, and I will welcome you on this next session. In the next session we will discuss about the mechanism of action of insulin, that is the molecular mechanism of insulin, and how insulin controls our blood sugar level. If we then get time, then we will be discuss the pharmacokinetics and dynamics of the insulin. Okay, thank you.